Greetings, good morning, good afternoon, good evening from wherever you are dialing in today. We're very excited to have you. Today we have a webcast on surveillance of antimicrobial resistance emergence in the environment. We're really excited to have everyone dial in and join this webcast. My name is Lola Olabode. I'm a research principal with the Water Research Foundation. So we have a great lineup of two wonderful speakers. The first time that we're having a veterinary doctor actually lead one of our projects, and her team with also Dr. Ellen Gunyen. They're both from the University of Illinois in Urbana-Champaign, and we have a phenomenal moderator, co-moderator, Dr. Amy Pruden. And so before we get started, just a little bit of housekeeping items. At any point, you could use the button to send in your questions through the box, but we'll do a Q&A for 15 minutes at the end of the presentation. Since it's just 1 hour with two speakers, we have a full agenda. And you could download the slides now by accessing a link to the project page. The project number, for those familiar with our website, is 5182. This project is ongoing, but this is a very critical webcast to get utility interaction and partners engaged in antimicrobial resistance surveillance as we keep moving targets. And again, I turn to our wonderful speakers. This research project is a result of our research priority program under a subtopic called monitoring tools. So here at the foundation, we're looking for innovative, forward-looking tools to protect the environment and safeguard sanitation. At the end of the webcast, you could have a certificate of completion that will be automatically generated, and you could please contact Michelle Swazo for that. Her email is right on your screen. So please stay until the end to fill out a quick survey. So next slide. Again, without further ado, our presenters and our wonderful moderator, their pictures are up here. And you could always toggle along your slides to learn more about them. But I'll turn over the mic now to Dr. Amy Pruden, who is well-versed and a scholar in this topic area. So it's no surprise that I reached out to her to get your participation and interaction. Thank you so much. And Amy, over to you.
Thank you so much, Lola, and thank you all for attending today's webcast. I'm really pleased to have the opportunity to introduce our speakers today, who are the principal investigators of Project 5182. So you do have their bios to read more details, but just kind of a highlight. So our first speaker is Dr. Helen Nguyen, who I've known for many years, and she's the Ivan Raichev Endowed Professor of Environmental Engineering at the University of Illinois at Urbana-Champaign. And she is a recipient of many, many awards throughout her career, but most recently, she received an award from the U.S. mission to ASEAN as a Fulbright Scholar to Southeast Asia, and she got a grant prize from the Trinity Challenge in the UK, specifically to develop a technological solution to antimicrobial resistance at livestock farms in low and middle income countries, so very relevant to the webcast today. And she really is a renowned researcher on a number of topics and pretty much anything related to microbes and pathogens in environmental systems. And so she's worked on water reuse, food safety, impacts of extreme floods on pathogen spreading, pathogens in drinking water and distribution systems, and also water quality after natural disasters. And Dr. Nguyen will hand things off about midway through to our second speaker, who is Dr. Becky Smith. And I'm also very excited to see a doctor of veterinary medicine presenting today. Dr. Smith is an associate professor of epidemiology, also at the University of Illinois, and she's in the College of Veterinary Medicine and Health Innovations at the Carly, Illinois College of Medicine. And Dr. Smith received her doctorate in veterinary medicine and PhD in epidemiology from Cornell, from their University College of Veterinary Medicine. And she also has a master's in biosecurity and risk analysis from Kansas State University. And so Dr. Smith also works on a number of topics, especially on surveillance, forecasting, and control of infectious diseases. So we're very pleased to have you, Dr. Nguyen and Dr. Smith. And after your presentation, we'll look forward to some Q&A with our audience.
Dr. Nguyen, go ahead.
Thank you very much for the opportunity to present the work that we have been doing and supported by the foundation today. So I would like to go straight into the problem. The antimicrobial resistant AMR is considered one of the top global public health problems. It's not only about human health, but it's also about animal health, livestock, food security, and economic developments. So the agenda of today's is we will briefly present to you that this is a public health crisis that wastewater facilities can clearly help to control. And how to do that, we propose two solutions. One related to how to measure antibiotic resistant genes, and more importantly, that my colleague Becky Smith will take over, is how public health authority can use the data. Microbes are very smart. They know how to protect themselves. So they always develop resistance. That's just the natural systems. However, when there is too much antibiotic is used, then the timeline for resistance development reduced. For the first antibiotics to develop the resistance, it took 15 years. Recently, it took only 2 years. So 2 years is too short for the pharmaceutical industry, for the medical industries systems to cope with, to find another drug. It takes many, many years to develop a drug. So 2 years is very fast. And when we talk about antibiotic resistance, it is bacterial infections, but it's not only, it's not just simply infectious disease. It's also related to other cases that related to septic treatment, surgery, chronic condition, organ transplant, dialysis, cancer care. So no matter how much progress of modern medicine we make in those areas, if we don't solve the antibiotic resistant problem, then the progress will be reduced, because all of the patients under those conditions are susceptible to bacterial infections. It's not a surprise to see the report estimated that mortality from AMR is growing and maybe as high as. And here are the data for the US. In 2019, there were for nearly 42,000 deaths due to AMR and 173 deaths associated to AMR. And our population is around 333 million. So now it's below other diseases. However, on other diseases that rank on top of patients also very susceptible to bacterial infections. So contribution by wastewater surveillance could help to inform the medical doctors about resistant evolution before the resistance get to them by discovery AMR in wastewater. So from the pandemic, we know that we can very quickly assemble a wastewater surveillance network. And this network can assist public health in monitoring abundance and spread of the ARG of antimicrobial resistance genes of interest. Currently, the surveillance of AMR mainly done at the hospitals. What we are proposing now is turn wastewater again into the resource that help public health to solve this crisis. There are several methods to do it. The most classical method is to isolate bacteria and to measure antibiotic resistant bacteria. However, we cannot grow every bacteria. There are bacteria that we just don't know how to grow. So therefore, we will have to turn into PCR method, culture independent method. So many of us now are familiar with PCR also thanks to the pandemic. PCR are very good. It can quantify ARGs. It has very high sensitivity, and there are instruments available in many places. However, there is several downside of PCR. It may have false positive and negative. It has relatively low number of targets, and we have to decide what target to do before we do PCR, because we only find what we are looking for. So what to looking for is actually not an important question. Another method that has been very popular recently is metagenomic sequencing. This is a very comprehensive method, but may miss a needle in a haystack. So ARG are a very small but important proportions of the whole microbial communities. So metagenomic can probe a wide range of targets. Data can be updated when a new ARG can be found. However, it has low sensitivity for ARG, very difficult to perform, and we don't have yet the standard method. So thanks to the funding from the foundations, we develop a modified method of metagenomics so that we can pull out the ARG and sequence them. So we don't sequence everything, but we sequence only the gene that the ARGs. So we use a method called CRISPR's pull out. So CRISPR's is CRISPR cleavage is the same method that you heard about for gene editing. So this is this method has received Nobel Prize, and basically are changing medicine in a revolutionary way. So we are actually using exactly the same idea. So we are not modifying any gene now, but we used the mechanism of CRISPR cleavage to isolate the ARG before metagenomics. So the method has several steps, okay? So first we have DNA fragmentation from environmental samples. If we don't do CRISPR, this fragment will go to ligations and then PCRs. With the CRISPR, the first thing we do is we do the 5' phosphate removal. Basically, we block at those fragments so that it cannot do ligations. And then after that, we do a CRISPR cleavage to cut it at only the site that can be ligated and then do Illumina sequencing. The protocol of this method is already developed and posted. You can use that QR code and get to the protocol that we already posted. Okay, so we design and synthesize guide RNA that guide the CRISPRs to cut the gene to target 5,000 plus antibiotic resistant genes. So we removed DNA from human, animal, plant, anything that not related to ARG. So we target 5,000 plus. So this is something that even high throughput PCR cannot do. So there are several methods that we have to go through this one. So I just want to emphasize that we have a CRISPR-Cas9 cleavage reactions before ligations. And I will not go into step by step of this because there's a protocol that already posted online that you can follow. So to validate this method, we also created a mock community to confirm the method. So we see here, so we have CRISPR ARG cluster, so positive, negative, so we found in this mock community here, we found 123 true positive. So we also conducted whole genome sequencing of this mock community. So from the whole genome sequencing, we found 97 of the ARG. We found more from 25 ARG from BLAST, tool, and then one more from Sanger sequencing. So we only found one form positive, one among 123, and two form negative. So in this this graph here, the y-axis of the A finger is number of ARG detected. The y-axis of the B finger is number of ARG family detected. So many genes belong to one family. And then on the x-axis, then there are wastewater samples from Chicago area. The CRISPR method detected the genes that are colored in the orange color here. Right? So the orange color show you the number of the genes that detected only by CRISPR method. The overlap between both methods are in the pink color, and the regular method is the regular method only is in the blue color. So what we see here is that this method works much better than regular metagenomic library sequencing, especially for ARG in low abundance and high diversity. So this graph here shows the p-value on the y-axis and the read depth fold chain in the log 2 on the x-axis. So read depth, you can think about it like abundance, okay? So what we have here is CRISPR over regular. In this graph here that you see that all of the data that we do in this that we got here are plotted on the significant quadrangle of this plot. So that means that the CRISPR NGS method have much higher, significantly higher read depth compared to the regular NGS library. So this is actually actually another way to present the data. So the y-axis is the ratio of ARG detected by subsamples of CRISPR-NGS, meaning fewer read depth, and over ARG detected by regular NGS. So we can use fewer read depth from CRISPR-NGS to detect the same number of ARG. So what you see here, the number 1 on the y-axis. Okay, so anything above that means that we can use fewer read depth. So CRISPR-NGS also have a lower detection limit than regular NGS. So we plot the read depth on the y-axis versus qPCR relative abundance. After we did that whole CRISPR-NGS, we also did qPCR for 18 different genes. So, and then we make this plot here. So what we see here is that the detection limit that we can have here for the CRISPR-NGS is about 1 order of magnitude lower than the detection limit for regular NGS. Most importantly, this method reveals clinically relevant ARG in wastewater samples, and this ARG were not found by the regular NGS method. So, for example, here in this graph, in this table here, I highlight some families that the CRISPR-NGS found, but not the regular NGS. So, for example, beta-lactamase. 2 types of beta-lactamase, and the last one here, MCR5. MCR is a gene belong to colistin-resistant gene, which is the last resort of antibiotics. Okay. So this is the heat map of qPCRs for the sample that we analyze using the CRISPR-NGS method here. So the next one that I would like to show you here is how to get from the CRISPR-NGS to get to PCR targets. So qPCR is still a very important method. qPCR and now digital PCR, digital droplet PCR is very important. So how do we go from the CRISPR-NGS to PCR-based method? So we develop an algorithm, and again, we only post that online at GitHub. So this QR code here will bring you to that algorithm. So the criteria for the selection of PCR target or follow, it needs to be highly relevant to clinical drug use. They should be found in corresponding sewer shed and may have mutation in the sequence detected by CRISPR-NGS. So we have several steps that we have to go through here. The first 3 steps here of this algorithm related to get ARG and mutation sites from the CRISPR-NGS finding. And then I will walk you to the step 4 here. So the first 3 steps are related to the CRISPR-NGS result. The step 4 now is actually we have to come up with the antibiotics commonly used and search for the number, the ARO number in the database. So how do you get to this list of antibiotics? Basically, this is the collaboration with the local hospitals, public health authorities. So once we have that, we go to we run the algorithm, and we can design qPCR primer and probe using IDT primer quest tools. So we apply this algorithm to one wastewater sample with more than 1,000 ARG detected by CRISPR and GS. So the reason we pick one is because this one is the one that have the highest number of ARG. So we found 387 ARG belonging to 63 ARG clusters. And after that, we designed primer to detect this ARG after consultation with public health professionals. And for the next few slides, my colleague, Becky Smith, will talk about the result.
Thank you, Helen. So one of my rules for diagnostic tests in general and surveillance tests in particular is that unless you have an actual purpose for the test, that you shouldn't be doing it, that you need to have it change your action. So one of our questions with this is, how can we use this tool to change the actions in public health to guide public health to make better decisions, or to guide clinicians to make better decisions? So you already saw some heat maps that Helen shared with you as far as looking at the different genes and the different sites. So we'll be working with some advisory boards that we have, thanks to some CDC funding. We have advisory boards of public health and of clinicians who are willing to help us with this, to develop explanation and visualization tools. So here on the left, you can see a heat map where, again, you have location on the x-axis, but on the y-axis, instead of individual genes, you actually have groups of antibiotics that the genes make you resistant to. So you can look at any particular site and see how many particular antibiotics might be of concern in that area, or you can look at a particular antibiotic and see if there is a concern. So for instance, on the left here, you can see penicillinase and glycopeptides are pretty low abundance in the resistance gene, so we might not be too worried about those. Alternatively, you can see that the site labeled Champagne manhole, it has a lot of resistance to a lot of different antibiotics. That happens to be in the area where our students live. And from what my discussions with the head of our student health center, not terribly surprising. They do tend to treat themselves quite a bit with a lot of different antibiotics when they're feeling sick. On the right, we can break that down by class of antibiotic and particular genes, so we can see which genes are contributing most to the different antibiotic resistance. And then we can also take a single site, so this is one of our sites, and then we can look at the different antibiotics resistance over time, and we can see, for instance, in this site, that beta-lactamase abundance varies over time, but sulfonamide resistance stays up there a lot of the time. So these sorts of things can help guide public health decision making. And we'll be working with these advisory boards and with the State Department of Public Health and with other groups to develop some visualization tools. And the ultimate goal, luckily the CDC funding includes work with the Argonne National Lab with their Bacterial and Viral Bioinformatics Resource Center to develop public health dashboards where we can put these tools up on the dashboards so that public health agencies can work in a secure location where they can share this sort of data with their groups and hopefully be able to use these tools to make better decisions. Now, I know the Water Research Foundation is very excited because I am a veterinarian. I also do wear a hat of public health and also a hat of human medicine. So I wear a number of hats at this university. But I will say from a veterinary medicine perspective, this is actually really interesting because clinical veterinary medicine, small animal medicine, suffers from antimicrobial resistance concerns as well. And also we can use this to consider the difference in these genes abundance at sites with and without contributions from livestock facilities. For instance, one of the plants where we have samples has some wastewater from a pork processing plant. So we can look at what's the role of these different animal processing facilities in the emergence of these genes and in the abundance of these genes to look at that risk over time. But also this can be very helpful for veterinarians as well as for human clinicians of knowing what genes are out there and what do we need to be concerned about in this population. So I will pass it back to Helen for the final summary, and then we'll answer some questions. Helen, I believe you need to unmute yourself.
So this method discover more ARG in wastewater sample than regular NGS. We do not want this method to replace qPCR or digital PCR, but this method allows us to determine the target gene so that we can do it PCR. And I'm going to end our presentation with this call for actions here. All of our samples are from Illinois, mostly from Chicago and Urbana County, Champaign County. We would like to work with all of you to try this method for samples across the U.S. We believe that the finding will be very exciting. Yes, so this is all I have.
Wonderful. And thank you for saving plenty of time for questions. This is a very interesting perspective that you've provided, and I think there's a lot of excitement around CRISPR and what it can do across multiple fields. And now we see that it really has potential to help with needs in antimicrobial resistance surveillance. So we have a number of questions coming in. The first one relates to one that I had. So can you talk a little bit about maybe what kinds of biases there are? So it's quite impressive. You mentioned that you're able to capture over 5,000 ARGs. And these are ones from the CARD database. So I'm wondering if there is some aspect of selecting which ARGs are captured. And then also, how does it compare with some of the panels that are out there? So, you know, in the era of wastewater-based surveillance, we're seeing commercial sequencing panels that are marketed to capture ARGs. So it'd be helpful if you could speak a little bit about that.
Yes, I can start, and then Eugene, my student who's working on this method, can also add to it. So, so the CRISPR cleavage depends on the guide RNA. So we have to design the guide RNA first, right? For the CRISPR-Cas9, then it works when the genome have the PAM regions. Right? So that is the first criterion that we have. We have to right. So when we design, so basically we we design and synthesize 5,000 plus guide RNA. So once we have the guide RNA, then we can get to the CRISPRs, the CRISPR cleavage, the Cas9 cleavage. It's not a simple method, actually, so it does require time to develop and also to try. So every time that we do it, we actually have to run gel to make sure that that we have the product that we need before the sequencings. So to compare with the panels that out there, so we did look into some of that high throughput panel method. So we cover a lot more than the current panels. Eugene, would you like to add something?
Oh, yes. So I think the current panels are using hybridization method, which can target 3,000 ARGs and a few bacteria and something depending on the design of it. And we didn't do a direct comparison between our method and the hybridization panel, but there are a few differences between the methods. So the first thing is that the CRISPR-Cas9 method is much cheaper than that one because we don't need to synthesize probes and the biotinylated subsurface substrate to capture the probes. So for each library, CRISPR-Cas9 method add on around approximately $30 to it. But the hybridization method, based on my understanding, it's like at least $100 something per library. And most of the cases, we have to pull the libraries first before doing the hybridization capture to save the cost. And that will cause another problem is that if we have multiple different libraries, and some libraries have more abundant ARGs and some don't have that much, the libraries with fewer ARGs may be biased by hybridization capture after pooling. But our method will treat each libraries individually so that the bias will not as much as that one. And also a third one is that our adapter ligation is happened after Cas9 cleavage. So the ARGs will be preferentially ligated by the adapters. And for hybridization capture, the adapter ligation happened before the hybridization capture. So adapters still attach to all different kind of fragments. And after that, the hybridization capture panel will catch the ARG part and then do amplification. So I'm not sure how much this will affect, but it's also a difference here.
Okay. So a follow-up question while we're focused on technical aspects would be, with shotgun metagenomics, you have the opportunity to do some sort of where you, what's the word? Assemble, thank you. Where you assemble the reads to get context of the genes. Is that something that does get lost with this CRISPR-based method?
Yeah, I think that's the main drawback of our method currently. But one method to possibly capture the context is to use long-read sequencing.
Right.
So yeah, we already see some work that use CRISPR-Cas9 to cut the ARGs and then use long-read sequencing to capture the sequence between different ARGs. So yeah, that one, I think the main goal is not to target as many ARGs as possible, but like to analyze the context.
That sounds very promising. So I'll give you the next question, which is easy, and actually for Water Research Foundation is you showed some QR codes, and I think the audience is very interested in reading more. Is there a way to share those with the audience? Okay, so I think there'll be some follow-up with those. So good to see that interest.
Yeah, so we could get some of the QR codes and package it with the webcast deliverables. So that those will be available via the download on whichever QR code the team wants to share. Keep in mind, this work is still ongoing. So this is a special webcast to monitor and follow as the research goes along. And so Amy, I didn't want you to, I didn't know what the next question was, but I marked a nice urgent one from Cook County, just because I saw I coordinate wastewater surveillance activities. So you could still take your order, but I didn't want us to, we have plenty of time.
Yeah, let's do Cook County. Go for it.
Okay, so his name is Joseph Avinash from Cook County, the Department of Public Health in the jurisdiction where wastewater treatment plants are. And he would like to discuss with the research team if this could be applied to public health activities in his jurisdiction in Cook County. So I think that is a really nice last slide that you left in terms of what you're looking for, and you got a live interest right there. So do you want to ask or, you know, what is the, I guess, how we could be applied? It's not any specific than just interest.
Yes. And Joseph is actually with public health in the area where our samples that we were presenting come from. So Joseph, I'm glad you're on the call to see some of the results of that sampling. So I saw there was another question about what is the public health application? And that is a really good question. This is something that we're still working on. So our Public Health Advisory Board, we've been discussing possible applications. We've been discussing wastewater surveillance in general with this board. One possible application that we've come up with the board is in the course of an outbreak. It might be useful to have these wastewater samples both at the treatment plant and also at individual facilities of concern, such as hospitals, such as residential facilities, to be able to monitor that when we think the outbreak is over, is it really over? Are the genes still present in the wastewater? Is there what we call cryptic infection remaining in the population that we can't find in the people yet, but we know that it's there because it's in the wastewater? So that's been considered one of the possible applications. Another of our advisory board members has pointed out that unless we have a baseline, we won't know when there's a problem. So one of the ideas with this is that it gives us a baseline so that when we can see a spike happening in a particular resistance gene, that we will know that that is not usual and that there is something going on that we need to investigate. So those are two possible applications that we've come up with so far, but we're still working on it. And I definitely have copied your email address, and I will be in touch because our advisory board, we are always happy to add new people, and you are within our district. So I will be in touch.
Yeah, thank you for that. I think that's a big question, are the applications here. And a follow-up question, I think you both noted that, there's still value for the PCR-based methods. And so can you speak to what an action plan might look like in terms of screening with this CRISPR sequencing method and following up with ddPCR? What might that ideally look like?
So what we envision is that you don't need to do CRISPR's NGS on the time, and then you get the result. And in the discussions with public health authority, then you run the second product of the projects to find the to narrow down on the list of the target qPCRs or digital PCR.
So maybe related to
So
that,
for
I've
example,
seen, yeah. Go ahead.
Yeah. So for examples, currently in clinical setting, only MCR1 genes is monitored. However, what we found in the wastewater sample from Chicago is a different gene. It's a MCR5, and we also found other beta-lactamase genes. So the method, the method that we do here basically guide PCRs.
Okay.
Yeah, that is the reason why we created the second product of the project on the algorithm that we can go from the result here to the PCR design.
Got it. So I think this partially answers, there's been a few questions about cost and time of the CRISPR-based technique. So I'm wondering, well, first maybe answering those questions about cost and time, and then I think in a practical scenario, how you limit that. So it sounds like maybe the CRISPR method would be performed by experts of some sort, and then provide guidance then to, more
Exactly.
routine PCR-based use.
Exactly.
There might be an analogy to
envisions.
There might be an analogy to how hospitals currently monitor antimicrobial resistance, that usually on an annual basis, hospitals will create an antibiogram that summarizes all the resistance that they have noticed in the hospital over the past year. And then they monitor on a case-by-case basis. So it's a similar approach that maybe once a year or maybe just every few years you can run the CRISPR-based process to develop your PCR panel, and then you can continue monitoring with the PCR panel and updating as needed.
Mm-hmm.
So then when you do the full-blown CRISPR-based analysis, how long does that take, and what's kind of the magnitude of cost?
Eugene, you can answer this question. And
yes.
I know that we did that analysis.
Yeah. So we usually need, like, for example, 1 day to process samples and do DNA extraction. And we need maybe another day to prepare the guide RNAs. And on the same evening, we can start the overnight incubation of Cas9 cleavage. And the next day, we do the library preparation. So 3 days before sequencing, and then we submit it to the sequencing lab. They will run a few more days to get the results. So probably the optimal condition, it will take 1 week from the sample to get the sequencing result.
Yeah, that's actually not too bad. I know it takes my lab a lot longer than that to get our sequencing results. So just making sure I've touched on all the questions here. This one is interesting. It's from Jumana Al-Jafari from the National Institute of Standards and Technology, and it's great to have them in the audience, as we've talked about the need for standardization and methods that are comparable. I think this question's about extending application beyond just wastewater-based surveillance. So I'll just read this one. Can ddPCR be used to detect and quantify ARGs in stormwater? I published research on the contamination of stormwater with human and animal fecal matter in areas with separate wastewater collection networks. However, wastewater still finds its way to stormwater for applications involving the use of stormwater for potable and non-potable purposes. It'd be very useful. So I guess in general, beyond wastewater-based surveillance, what are some possibilities? Could we look at stormwater, surface water? Does it make sense to do that?
Yeah. As long as we have enough bacteria and DNA. So we are applying this method to other samples. So as long as you find enough bacteria and DNA, we will be able to do this method. So storm water runoff is have a lower concentration of bacteria. So you may need to do some concentration method to concentrate the bacteria first before DNA extractions.
I think some of it touches too on what Dr. Smith talked about, like, should we test it if we don't have an action in mind? So it sounds like there are potential actions here.
But there are potential actions here if you have stormwater that you're concerned about because it, for instance, came off of some livestock facilities or came off of a site where you're worried, then this would be a good method for monitoring, is the water safe?
Or getting into beaches or something.
Yes, exactly. Beaches, swimming areas. Yeah. And that is definitely one purpose that the Public Health Advisory Board was interested in, is monitoring for safety after an event of concern.
Yeah, and I think it's analogous to, beach and recreational water monitoring. They need a quick answer of, what do we do here? Can people swim or not? Right.
Yes. And I can actually see a purpose within the livestock industry of monitoring surface water safety for the animals. There's a lot of livestock facilities have surface water to provide to the animals for water. And if the water becomes contaminated because the neighbor has spread manure on the field or is spreading waste products on the field, this would be a way of monitoring for the farm to say, should I be allowing my cattle to drink this water or should I be blocking it off?
So as we're talking about sampling, expanding beyond wastewater, an important part of the protocol is sample volume collection, et cetera, extraction kit. So one of the questions is about whether you can use the same sampling method for both CRISPR-ARG and the PCR method, sampling, extraction, et cetera, or would they have to be different pipelines? And then there was a similar question about, does it matter if you do digital PCR or quantitative PCR?
So sampling in the same way, like I mentioned before, you just need to have enough bacteria and DNA. Digital PCR, so we plan to also try digital PCR. It's just more expensive. So that's why we didn't try it yet. But that is feasible. We are deciding it now, like what gene we want to target with digital PCR. Yeah, so for the cost, the reason that we didn't do long-read sequencing, also we try to use NGS because it's cheaper. But we can also move to the long-read sequencing. We do that with other things, and it's an expensive thing. So the Cas9 is expensive. So therefore, we don't want to go with more product that very expensive.
So we have another question that is from Gwinnett County. This is from Robert Harris. And it is there a difference in turnaround time from CRISPR-ARG versus qPCR? And then he also asked about the difference in sampling methods. But what about the turnaround time? So some people say you can do qPCR in a day. Would you agree with that? So we heard that CRISPR-ARG takes a week.
Yeah, because it's also the sequencing time, you know, so like preparing the library take 3 days. And then you have to wait for sequencing, you know, and they don't sequence. Usually you don't sequence yourself, you have to send somewhere. And we have a very good sequencing facility on campus. So that's why it takes only like a week. And we actually talk to them like when you're going to sequence, and then we back out and know when we have to send them the samples. So qPCR is definitely much faster too. So we are not saying this replace qPCR by any means. It just helps us find the qPCR target. And it's not a method that we want to do it routinely. Like anyone screening makes sense.
I think we've touched on most all the questions. Lola, are you seeing any urgent ones we missed?
Maybe just a clarification, since we're asking for participation, is it's from Anna Marotra, who leads the public health surveillance at the Water Environment Federation. And it's to define, so she has a lot of access to utilities. So if you could just define large size, mid-size, is it in terms of average daily flow or population served? I know you gave examples in that table, so I was assuming population, but...
Yes, it's in terms of populations, yes.
Okay.
Yes,
so that helps.
I saw there was another question about comparison with the clinical surveillance data, and that is specifically why we want a range of size of population served, because the next steps will be comparing these results to hospital data and to public health data. And we need to see if the size of the population served influences how well this represents what's in the clinics.
And then my darling colleague Grace Chang is asking a question about technique. So considering the approximate 7-day turnaround, I think we've already discussed this. I think she's looking for an answer about the suitability of in emergency responses. Is the technique suitable if it's a 7-day turnaround?
So
I guess it'll be site-specific,
like
disasters,
situations.
Yeah, so it's
science-specifics, you know. So I think if we can...
Okay. Please go ahead.
Yeah, so for emergency, I would do in parallel qPCR and this method, because qPCR is actually very, very good first. And then at the same time, taking a week to do this method and see whether or not we have to refine the PCR target.
I think there's one more final question that I think you're looking at the same one, Lola. This will be a great closing question. It's from Steve Rode at MWRA. Yes. So how are you planning to solicit facilities to contribute samples? So you mentioned at the end that you'd love to pilot test this across the country. So how will folks hear from you to do that?
So I plan to work with Lola on this with the foundations on this part here. So definitely get in touch with us, and we can discuss further. And I will definitely keep Lola in the loop for this, because the foundation actually provides supplemental funding for extending this work beyond Chicago.
Okay. Thank you so much for that. And we're at the top of the hour. I think that last question is really key in terms of logistics and how people can get involved. I did look at one of our other programs to see if we could support at least the analysis and the logistics of how many utilities we get. So there are different ways we could do this. Utilities can directly contact Ellen and Becky, or you could go home through WRF for like I don't want to say more structured, but we'll be able to keep track, because I don't think we're looking for every single utility. They've given like a nice snapshot of the coverage. Again, we have extensive utilities. I don't want to exaggerate, but maybe 10 in that Illinois area, small, medium size, but like Ellen said, we want to expand outside of Illinois to kind of show the broad applicability of this surveillance technique, especially since we're constantly moving targets on what surveillance, monitoring tools are more appropriate as we prepare for like the next health crisis situation, or even endemic ongoing situations. So with that, I think we can wrap up our webcast. For the questions that you feel like did not get answered, or you're still looking for a response, we will share that with Becky and Ellen and Yuqing to respond to and package that, and also have that on the website. So we will definitely respond to everyone. You're encouraged to contact me, the manager for this, and Dr. Amy Prudence. So like I said, this is ongoing, and we have some exciting activities at the foundation to continue in this space of AMR surveillance, quantification, risk assessment, and other water matrices. So we're expanding, and we're going to be here for a while. So I want you all to, just one last logistical housekeeping item is to fill out the survey if you have a minute before you drop off. Michelle, if we could put the survey slide, or you could click on your survey slide, just to help us with what kinds of webcasts. I know a lot of people are used to waiting till the end of a project, but if you're interested in some ongoing preliminary findings, activities, let us know. So that survey should have popped up. I really want to know if you would like to have something like this again, rather than waiting till the very end. I think we're scheduled, depending on the kind of participation we get, for another 1.5 years. Is that correct, Becky? About 1.5 years. And then we have a new endeavor that once we get started will be 3 years. So we're very much going to be in this space. So now we're building our community. So with that, We look forward to hearing from you all for your interest. It will certainly help me with my own internal pitch to be able to support this activity. If I could say, ooh, everyone that came on the webcast is interested in supporting, that would be a powerful statement. But let us know, and enjoy the holidays and stay safe and warm. Thank you very much.